Good morning, ladies and gentlemen. Welcome to the SAREUM Holdings PLC annual results investor presentation. Throughout this presentation, investors will be in listen-only mode. Questions are encouraged and can be submitted any time by the Q&A tab situated on the right-hand corner of your screen. Simply type your question and press send. The company may not be in a position to answer every question received during the meeting itself. However, the company review all questions submitted today and publish responses where appropriate to do so. These will be available via your Investor Meet Company dashboard, and you'll be notified once they're ready for your review. I'd also like to remind you that this presentation is being recorded. Before we begin, we'd like to submit the following poll. I'd now like to hand you over to Dr. John Reader, CSO, and Dr. Tim Mitchell, CEO. Good morning.
Thank you, Paul. Good morning, everyone. My name's Tim Mitchell. I'm CEO and a co-founder of SAREUM. Also presenting today is Dr. John Reader, Chief Scientific Officer and co-founder. And thank you for joining us as we present our progress and financial results for the last year. Before we start, I need to show you this disclaimer. So as a reminder then, our business model is to do the early stage design and development of new drug candidates and then license them on to larger pharmaceutical companies for further clinical development and licensing. We've developed an exciting pipeline of internal and partnered pipeline assets that address emerging high-value disease targets, which we'll describe in more detail later in the presentation. Our business model has been validated by our CHK1 kinase cancer program. Working with the Cancer Research UK funded organisations, we developed SRA737 and took it into phase 1 studies before it was licensed to Sierra Oncology in a $300 million deal. Internally, we're focusing on our TYK2 JAK1 kinase inhibitor programs, SDC-1801 targeting autoimmune diseases, including of particular current interest, the cytokine storm that's often the cause of death in COVID-19 patients, and SDC-1802 targeting cancer and cancer immunotherapy. We're a very experienced team of management directors and advisors with a deep understanding of drug design, development and commercialization. We've been listed on AIM since 2004, and as you may have noticed, our share price has risen considerably since we last met here, such that our market cap is the order of £220 million, which I hope you'll agree is a significant increase in value from the £21 million cash we've raised to date. For those of you not so familiar with us, let me introduce you to the board then. So from the top, Stephen Parker, our chairman, has a long career in healthcare and pharmaceutical corporate finance and investment. I've worked in large, medium and small pharmaceutical and biotech companies for over 30 years now, with over 17 of those being at SAREUM. John Reader has a long research career in biotech, both in the UK and the US, and likewise has been at SAREUM for 17 years. Michael Owen, non-exec director, was biopharmaceuticals head of research at GSK before leaving to be the co-founder of Kymab. Mike has a long expertise in immunology, which is particularly useful to us. Clive Birch, also non-exec director, has a career in audit, specialising in early stage healthcare companies, and is a great asset with our compliance and corporate governance. This is a snapshot of our research pipeline. At the top, we have our TYK2 JAK1 programs, with SDC-1801 nearly completing its preclinical studies in autoimmune disease, and the COVID program running alongside it. SDC-1802, the cancer and cancer immunotherapy program, is a little further behind in its preclinical development. SRA737, the molecule licensed to Sierra Oncology, has completed 2 phase 2 studies, one as a monotherapy and the other in combination with low-dose gemcitabine. As well as the gemcitabine combination, there's an emerging body of preclinical data on combinations with other targeted cancer therapies, such as BET, PARP or WEE1 inhibitors, and also cancer immunotherapy. And Sierra have indicated their intention to continue SRA737 development in one of these combination settings in the first half of next year. Moving on to the highlights we reported for the last year, we've made substantial progress despite the challenges of COVID-19. So in SDC-1801 for autoimmune and severe COVID, we're nearing the completion of the preclinical phase with the intent to file CTA, that's a clinical trials approval, in mid of next year, and start phase 1 clinical trials using a higher performing capsule drug formulation shortly after that. For COVID-19, we've received a 170,000 grant from UK Research and Innovation to explore the potential against severe respiratory inflammation caused by COVID and other viral infections. And we've reported very encouraging results from the preclinical studies on this, demonstrating cytokine regulation in several disease models. For SDC 1802, we're currently designing translational studies to define the optimal cancer application prior to completing the toxicology and manufacturing studies required to file the CTA for that program. And our intellectual property portfolio has been strengthened with 2 US patents for 1802 being granted, and also the publication of a solid and crystal form patent for the TYK2 program. For CHEK1, the license agreement between Sierra and our partners, the Cancer Research Technology Pioneer Fund, was amended last year, which we believe will expedite further development. And actually, Sierra have made a number of announcements on the potential future of SRA 737 recently. So they've in-licensed a BET inhibitor from AstraZeneca, which is now known as SRA 515, and they're talking about potential combinations with that molecule, plus in other scenarios. And so we believe Sierra will be in a position to continue clinical trials in the first half of next year. On the finances, we raised £2.37 million in June and reported cash in the bank at the end of the financial period as £2.7 million. We reported a loss after tax of 1.5 million. That's increase on previous years, mainly due to the extra expense of the research involved in these preclinical stages. And then the balance sheet was further strengthened post-period end by a further 2.18 million being raised, so making a total of 4.6 million raised through a number of share subscriptions and warrant exercises. So if we now move on for a more detailed look at our research programs, I'll do an introduction and then hand over to John. TYK2 and JAK1 are members of the JAK family of cell signaling enzymes, which are important for maintaining a healthy immune system, and as such are of targets of great interest to pharmaceutical companies. There's been good clinical validation for TYK2 and TYK2 JAK1 from BMS and Pfizer in diseases such as psoriasis and psoriatic arthritis. And these companies are completing further phase 2 trials in other autoimmune diseases, including lupus, ulcerative colitis, Crohn's disease, with readouts expected throughout the next few years. There's also good scientific evidence for the role of TYK2 in modulating the severe inflammatory response and symptoms arising from coronavirus and other viral infections. And several JAK family inhibitors are being tested in the clinic or being prepared to test it for COVID-19. The market size for JAK family inhibitors currently stands at around 18 billion, but has been projected to rise to 320 billion by 2026. And BMS are forecasting 4 billion sales for their TYK2 inhibitor deucravacitinib in 2029. I'll now hand over to John to update you on the status of our programs.
Thank you, Tim. So the JAK family kinases transmit extracellular signals from cytokines through to the STAT transcription factors, which go on to produce a wide variety of gene products. The cytokine binds to its receptor, and each receptor is associated with one or more particular JAK kinase family members. And some of the key cytokines, their receptors, and their associated JAKs are shown here on the slide. As you can see, TYK2 and JAK1 signal all of the therapeutically relevant cytokines shown here, except for EPO, which is involved in red blood cell formation. And of course, we don't want to inhibit that pathway. You can see that's dependent exclusively on JAK2. It's JAK2 and JAK3 inhibition that are associated with the FDA black box warnings for serious infections, lymphoma, and thrombosis that have been issued to the JAK1, JAK3 inhibitor Xeljanz, formerly known as tofacitinib, and the JAK1, JAK2 inhibitor Olumiant or baricitinib. So we believe that there are significant and clear opportunities here for SAREUM. We're focused on our selective and potent TYK2 JAK1 inhibitors. There are currently no TYK2 selective products on the market, and there are no selective TYK2 inhibitors in clinical trials for cancer, so that gives us a first-in-class opportunity with SDC 1802. We're developing two distinctive TYK2 JAK1 kinase inhibitors. Both are potent inhibitors of TYK2 and JAK1 and have good selectivity over JAK2 and JAK3, and importantly, the rest of the more than 500 kinases that make up the human kinome. In our mouse disease models, we dose orally twice daily, but based on our modeling, we believe we'll be able to dose once daily in any human studies. We've noted the potential of our TYK2 JAK1 inhibitors to treat the cytokine storm that can be fatal in many COVID-19 patients. We've been encouraged by the promising results achieved from our UKRI grant-funded preclinical study, and we're exploring ways to progress this into clinical studies. There's also potential application for our molecule in long COVID, although I would say the science is less well established there. But obviously we're keeping a very close eye on those studies as they emerge. And then we have a strong patent portfolio protecting our products in the major markets, and that's been exemplified by our recent US patent grant announcements. Our internal focus is on completing the preclinical studies on SDC 1801 TYK2 JAK1 inhibitor so that we can start its clinical development. To this end, we've recently appointed consultants to help us develop the clinical plan and to give us some guidance as to the optimal autoimmune disease indication that we'll use for our first studies in patients. We've presented these programs to many potential pharmaceutical partners, and we'll be looking to strike commercial licenses during the early clinical stages onwards, bearing in mind the risk-return funding requirement balance of an early deal versus a later stage licensing. The slide here shows a roadmap to starting the studies. So for the remainder of this year, we'll be completing the toxicology studies on SDC 1801 required to file an exploratory CTA while we continue to develop the plan for the clinical studies. In the first quarter of next year, we expect to complete GMP manufacturing of SDC 1801. So this is the actual production of the molecule itself on large scale. That's in progress as we speak and will be completed early in 2022. And then in Q2 next year, again, these studies have already started, but we would aim to complete the development of the capsule formulation for the SDC 1801 drug product. It's a more advanced formulation than the powder in bottle formulation we'd originally envisioned, and it does take slightly longer time to develop, but we believe that it gives us some clear advantages, a better chance of a meaningful and successful clinical trial result. Once preparation of those capsules is complete, we'll be able to file our application to commence clinical trials. We expect the trial to commence in Q3 next year, starting with a phase 1A safety and dose ranging studies in healthy volunteers. And then alongside that, we'll undertake some further toxicology studies so that we can run a phase 1b trial in autoimmune disease patients. And we'll also be looking for further opportunities to progress SDC 1801 in severe COVID-19. So a phase 1a study that I've just described in healthy volunteers will set us up nicely for trials in COVID patients. And we'll also be exploring further funding opportunities for this from schemes such as the UK government's Therapeutics Task Force and the Agile Clinical Platform. Moving on to SDC 1802, the molecule has shown good efficacy in a number of cancer disease models, including kidney, colorectal, melanoma, and B-cell lymphoma. There's strong evidence that it exerts its effects through an immunotherapeutic mechanism. We've seen positive results in cancer models in immunocompetent mice when dosed orally as a monotherapy, or additive effects when used in combination with chemotherapy. Recent patent grants, as mentioned, have strengthened our IP portfolio in the major territories, with protection for the composition of matter, that's the molecular structure of SDC 1802, and also protection for its use in the treatment of certain cancers. Currently in progress, we're carrying out translational studies to help us understand where best to use SDC 1802. The studies will help us to understand more precisely the mechanism of action of a molecule, and that in turn will guide our selection of a patient population for any subsequent phase 1 trial. So for example, we might be selecting to run a trial in a specific disease such as colorectal cancer, or it might be in a population of patients with cancers at different sites, but all with, for example, an elevated interferon gene signature or other biomarker. We're aiming to get substantially down this path before completing the toxicology and manufacturing studies required to take SDC 1802 into the clinic. I think it's worth mentioning that our experience in the development of SDC 1801 will be very helpful in the development of 1802. So, for example, the synthetic path is very similar. The first 2 steps are identical. So what we've learned in the development of 1801 will really help us in development of 1802. So moving on to our partnered candidate SRA737, as well as completing 2 phase 2 clinical studies in the hands of licensed partner Sierra Oncology, SRA737 has shown exciting potential in an increasing number of preclinical combination studies. The one I'm showing here is the triple combination of SRA737, low-dose gemcitabine, and an immune checkpoint therapy. And you can see an absolutely spectacular result, 100% tumor regression in this preclinical model of small cell lung cancer, a very difficult to treat lung cancer. As discussed also, though, there have been very exciting preclinical data showing its potential in combination with BET inhibitors, PARP inhibitors, DNA, POLB, and WEE1 inhibitors. So we're really excited about the potential for Sierra to start some additional clinical studies, hopefully as soon as possible next year. Okay, that's the end of the technical part of the presentation. So I'll hand back to Tim now to take you through the rest of the presentation.
Thank you, John. I'll finish this part of the presentation with the current clinical development status of SRA737. Then we'll go through the financials and look ahead to see what we might expect from the next year, and then finish with the answers to your questions. So as I mentioned earlier, towards the end of last year, we announced that the license agreement between our partners, CRT Pioneer Fund and Sierra Oncology, had been renegotiated. So it reduced the total amount of milestone payments, but introduced a new $2 million milestone payment. So this would be payable on the first patient dosed in any new clinical trial. SAREUM still remains eligible for its 27.5% share of the economics of the deal. So that would result in a $550,000 payment being paid to SAREUM on that next milestone of the first patient dosed. And our total share of future milestone payments is getting on for $80 million now. So recently, Sierra has indicated that it will indicated that it will continue the clinical development of SRA737 next year. They've licensed a BET inhibitor from AstraZeneca and have presented opportunities for clinical studies in combination with SRA737. They've also noted the potential combination studies with SRA737 in PARP-resistant pancreatic cancer, in addition to the previously described potential for clinical studies of 737 alongside other anti-cancer agents, including immunotherapies and PARP inhibitors. So any one of those clinical studies would trigger this new milestone payment on first dosing. We look forward to providing updates as and when Sierra announces their further progress. So if we turn to the finances that we reported a week or so ago, in the income statement, we see the UKRI COVID grant of 171,000 in the other operating expenses line. Again, as I mentioned earlier, the operating expenses have increased quite a bit from last year, but this reflects the high cost of the preclinical studies that we've been undertaking. We also see the R&D tax credit of £218,000, which we will receive as cash, usually early in the new year. In the balance sheet, we see the cash balance of 2.7 million, most of which arose from the two subscriptions that we announced in June. That was for a total of 2.4 million from a high net worth individual. This has been further strengthened during July and August by two further placings to high net worths, totaling 2.2 million, such that we had a quite healthy cash total of 4.5 million at the end of September this year. So if we look forward to what we might expect to achieve during the rest of 2021 and into 2022, for TYK2/JAK1 then, we're looking to complete the SDC 1801 preclinical tox studies by the end of the year. If you remember the timeline that John presented earlier today, then we're looking to file for the clinical trials approval by Q2 of next year, and then start the phase 1A stage of the clinical trials shortly thereafter in Q3. We hope also to be reporting updates from the UKRI grant-funded COVID-19 research and the next steps there, and updates on SDC 1802 as we progress that through preclinical. For the 737 CHEK1 program then, so we're looking to see further clarity from Sierra on future development plans, so I suppose in particular which particular combinations and which particular cancers they're going to be targeting. So there's the potential for advancement, as we mentioned earlier, of 737 into further clinical trials. So these could either be in combination with the triple combination with low-dose gemcitabine and immune checkpoint blockers, and/or the combination with the BET inhibitor SRA515, or a host of other potential combinations such as PARP and WEE1 inhibitors. And as I've just said, the milestone payment, that's the $2 million, of which we'd receive 550,000, would be payable on the first patient dosed in any one of those trials. So we're expecting to report important preclinical and clinical progress during 2021 to 2022 from the continued development of all these programs. Okay, so that's the end of the presentation part of the presentations, and thank you very much for listening in. Shall I hand back to Paul to start the Q&A session?
That's fantastic. Thank you very much indeed. Tim, John, thank you indeed for the presentation today. Ladies and gentlemen, do please continue to submit your questions using the Q&A tab situated in the right-hand corner of your screen. Just click Q&A, scroll to the bottom, type your question and press send. But just while the team take a few moments to review those investor questions submitted already, I'd like to remind you the recording of the presentation, along with a copy of the slides and the published Q&A, can be accessed via your investor dashboard on the Investor Meet Company platform. I'd also like to remind you that your feedback is important to the company, and immediately after the presentation has ended, you'll be redirected for the opportunity to provide your feedback in order that the company can better understand your views and expectations. Tim, John, we received a number of pre-submitted questions from investors, together with questions submitted during today's presentation. If I may just hand back to you to run through those pre-submitted questions, and then click on that Q&A tab, and where appropriate to do so, read out any of today's questions and give your responses where appropriate to do so.
Thanks, Paul. Let me just check now. My mic is on. Great. Yes. So thank you very much for your questions, both during the session and also the ones pre-published. So we've had a lot of questions posted prior to this meeting. What I've done is group them into the various categories they fall into. So if I may, I'll start by answering each group of these questions. So there've been some questions regarding our cash runway. So have we enough cash to get to the direction we need to get to? Is the plan to take all our compounds all the way? I presume that means to market. And then also regarding cash runway for our current projected and planned activities, how much runway does our current cash in hand give us? Is it sufficient for phase 1, phase 2 of 1801 and 1802? So the new funds raised over this June to August period, so that's the 4 and a bit million, are anticipated to allow us to execute our near-term strategy of advancing SDC 1801 into clinical trials and completing the preclinical work on 1802. As we've mentioned in our full year results, we'll require further funding to take these programs any further than that. We don't have any plans to take our compounds all the way to market. We're looking to out-license them at the appropriate stage, but certainly before we think about getting the compounds approved for marketing. Moving on then, there's some questions regarding licensing and partnering. There's been talks of partnering for a considerable time now. Is this because we're expecting a better financial agreement, or the lack of partner being able to invest? Could the board expand on the partnering strategy? What's the strategy for when therapies will be licensed and the type of partners selected? Could you clarify if the intention is to go forward in partnership, or as SAREUM is to be bought over? So if I can answer those then. So we continue to engage with a number of potential licensed partners, but can't provide any further details as to the status of these interactions. Of course, we'll communicate any developments that will require disclosure to shareholders. Our business model and expertise are aimed at designing and discovering new drug candidates and developing them through early development, and then realizing value for shareholders through out-licensing them to partners. As we previously said, we'll aim to out-license 1801 and 1802 at the appropriate stage, and the revenues generated from that would enable us to look for other opportunities and repeat this process. We keep an eye on attractive programs to which we can add similar expertise and value. And as we noticed in the presentation, we continue to assess the potential returns, funding requirements, and risk of licensing at a later versus an earlier stage. We continue to keep our interested counterparties updated with our progress, and continue to be prepared to discuss any form of transaction where we believe the terms would be beneficial to shareholders. We have quite a lot of questions regarding the recent publication of a solid form patent. So could the crystallization techniques covered by the patent be applied to other compounds such as FLT3? Regarding that patent, does the discovery enable you to address formulation problems with Aurora FLT3 program? Several questions along that line. What are the implications for competitors using the formulation process in the patent application? And what are the timescales on the reveal of? It's called the secret patent. I think that was the status of the patent pre-publication and potential licensing partners. There are 3 other individuals as co-applicants named on the patent. Would they be entitled to any percentage of a revenue share of any license revenues? And then a further question on which particular program it applies to. So in answer to those programs, then, so this patent was recently published on 14th of October, and as we noted in our full year results, it describes the solid and crystalline forms of a TYK2 inhibitor. This type of solid form patent is standard pharmaceutical industry practice, an important step to fully protect the intellectual property surrounding the company's research programs and to extend the patent life of its portfolio. So the techniques used here can't necessarily be applied to the Aurora FLIP program. They were specifically aimed at the TYK2 program. They may or may not be applicable to the Aurora FLIP3 program. I'd be surprised if they were useful there. So we don't believe there's any formulation information in the patent that could be used to advantage by our competitors, and we retain our IP lawyers and advisors, as you'd expect, to make sure that we don't give out any unnecessary information that could be advantageous. So although there are co-applicants named on the patent, it's wholly owned by SAREUM, so the co-applicants are not entitled to any revenue share. If I move on then, there's some questions regarding the capsule formulation of SDC 1801. So yeah, the CTA for 1801 has been delayed over the last few years. When we gave a communication in previous RNSs, did we not consider it would be better to wait for capsule formulation? Why are we adding delays by waiting for capsule formulation? So our decision to go down this route has been influenced by outcomes and ongoing preclinical studies and formulation and manufacturing work we've done to prepare SDC 1801 for clinical trials. This data, alongside the additional funding received during the year, gave us an opportunity to look at ways to add value to our programs earlier in the development process. So the capsule formulation was something that we'd have to do anyway at a later stage, and we thought it was in our best interest to go straight to that formulation rather than a simpler type of formulation to start with, and then move on to the capsule formulation. We have some questions regarding SRA 737. So how confident are you that Sierra will progress 737 in the next 12 months? Are we excited about the range of solid tumour cancers that the CHEK1 inhibitor could potentially treat? So we can only go by what Sierra have reported in the public domain, but our understanding is that Sierra intend to recommence clinical trials with SRA 737 in the first half of 2022. And yes, we see the number of potential combinations with other inhibitors and potential clinical settings as a great advantage for SRA 737. So related to 737 and partnering questions, there's a question about what lessons we've learned from the previous licensing deals with 737 and FLT3 and Aurora. So yes, there is learning to be had from these previous agreements, and we're putting these into practice in our preclinical studies to ensure these and formulation and manufacturing processes and clinical trial plans are well thought out and robust, and position our programs in the best light for this. So there's a number of questions regarding 1801 and COVID. So given the promising results for 1801 with COVID, why has 1801 not been admitted to Agile, and what's the current status? If the government's Agile program is used to progress, do you foresee any potential drawbacks, i.e. losing control of the program? Can we give more clarity about the timing of a COVID-19 clinical trial? I'll be discussing further work on COVID with the governments in the other countries, and given the possible connection between long COVID and cytokine response, have we looked at the role of 1801 for long COVID? So taking those questions then. So Agile is a clinical trial platform. So we need to complete our preclinical studies on SDC 1801 before considering Agile or any other clinical trial funding programs. Our understanding is that we wouldn't lose any control of the program if we were successful in obtaining Agile or similar funding. The phase 1A clinical dose ranging and safety studies, as John mentioned, that we plan for 1801 will be applicable to both COVID-19 and autoimmune diseases. We'll continue our dialogue with experts and consultants, read the funding and location for further COVID-19 studies, and take their advice as we see fit. And we're aware of the report suggesting elevated cytokine levels as one of several mechanisms responsible for long COVID. So we continue to monitor the literature and consider how we might experimentally demonstrate the benefits of a TYK2 inhibitor here. Another proposed long COVID mechanism connects it with elevated autoantibodies, which are also observed in lupus patients. And we have preclinical data to show our TYK2 inhibitors reduce the levels of these autoantibodies. So there's another angle into long COVID that we could potentially explore. We have a number of questions regarding the subscription by the high net worth individuals. So were we approached by them, or did we approach the individuals? Was this part of a defined fundraising strategy? So I think in answer to all those questions, we really can't disclose any details on any investors or their holdings unless they're above the 3% level where we're required to report, or their company directors and their close associates. And then finally, from the pre-submitted questions regarding Peel Hunt, could you outline the reasoning behind the appointment of them as co-brokers and the timing? So appointing Peel Hunt as co-broker gives us the opportunity to be given specialist life science analyst coverage, plus also an additional route of access to institutional investors. So those are the pre-submitted questions. I think we've got some time to run through some of the ones submitted during the meeting. Give me a moment while I look through. So there's a question about how many parties. I think this is licensed parties that we're interested in. I think I've covered that in some of the pre-submitted questions. In that we can't really disclose too much information about who and how many partners we're talking to. There is a question about the split in R&D expenditure between autoimmune, COVID, and 1802. I think as you've seen from the presentation, right now the autoimmune and the COVID programs are running in parallel, and we'd expect them to receive the lion's share of future funding. But there's some important progress from 1802 that will also be funding as well. But I think it's generally the further down the research track you go, the more expensive the experiments are. Hence the greater share of R&D expenditure being on the 1801 program. There's a question about how many TYK2 candidates the market can absorb, and then some related questions on capsule formulation, and then also TYK2 in cancer. So let me take those one by one. So I showed earlier a slide on the market size. So projected market size of the JAK family inhibitors of 320 billion, with BMS projecting 4 billion for their deucravacitinib. So I suppose you could almost do the maths on that, but of course that doesn't really cover individual. That covers JAK family as a whole rather than TYK2 in particular. I think where we are on that is that there's such a wide range of diseases that a TYK2 inhibitor could potentially be a useful treatment for. That there is quite a large market size, and we're not concerned about not being first to market here. I think I've covered off the capsule formulation route in that we'd be ending up with a capsule formulation for later stage clinical trials anyway. So that's that's sort of slightly unrelated to what the market would accept, because say that's something we'd have to do. And then in terms of the TYK2 and cancer indications, I think it's really a lack of understanding as to the precise role of TYK2 in cancer, and that's something that we are trying to understand in the translational studies that John described earlier. There's a question on the capacity of our CRO partners. So we've noted several times during the last year or so that our CRO partners are very busy, and we're not, well, it's not necessarily a capacity issue, it's just the lead times for starting new experiments seems to be getting longer and longer. So we just need to make sure that we book our slots in good time. But once our experiment's starting, we're not really seeing any delays in them being completed. There's a question on why Sierra haven't paid any milestone payments, and that's because the completion of the phase 1 and 2 trials weren't a trigger for milestone payments. So it's not like they've ducked anything or in breach of any agreements. The stages that they completed weren't triggers for the milestone payments. We have a question on the read across we can make from BMS's published TYK2 safety profile versus our compounds, and I think we're very encouraged by the good safety data that BMS are receiving from their TYK2 deucravacitinib programs. I suppose there's two reasons why two broad reasons why a novel compound can fail in clinical trials for toxicity type reasons. So one is that the inhibiting the target is going to give rise to toxic events, and it looks like that isn't the case from BMS's data. And then the other reason is the molecule has side effects on other targets, for instance, and that's essentially what we have to prove when we do our safety studies, particularly in the clinic. I'm just moving down. Then I think a lot of the questions have been covered off, and we'll group those in the other questions that we've covered previously in the answers we published. There's a question about are we releasing any preclinical data for our TYK2 compounds to the market? So we'll announce progress, I suppose, as part of our general reporting. So during the various periodic statements that we make, such as we'd expect to do a pre-AGM statement. We'll do a first half results announcements early next year, where we'll note the progress that we made then.
Tim, John, I think you have covered off an awful lot of questions as you've gone through there, but I think if there are any further questions that you can address, of course, the team will be able to review all questions that are submitted today, and we'll publish responses where appropriate to do so, unless there's anything else you want to address now, Tim.
I'm just looking through them. I think most of the ones in the pre-submitted questions. So forgive me if I've skipped over something that isn't quite that hasn't been covered, but it says as we said, we'll respond to all questions in the answers we publish post meeting.
I think then on that basis, Tim, if I may, and just say thank you very much for your presentation today, and along with John, and for answering so many questions. Of course, as Tim just said, we'll get some responses to those other questions that come through, and any further questions that are submitted by you, the attendees. So thank you again for submitting so many questions. Can I please ask investors not to close the session, as you will be automatically redirected for the opportunity to provide your feedback in order that the management team can better understand your views and expectations. This will only take a few moments to complete and is greatly valued by the company. On behalf of the management team of SAREUM Holdings PLC, we'd like to thank you for attending today's presentation. That concludes today's session. Thank you very much, and good morning to you all.
Thank you, everybody.